And then we'll also need to post it on Facebook.
Yes.
Yep, it is setting up the stream right now, so we'll be good to go in a moment.
Can you put the link to the stream like in the Zoom chat, maybe?
Here we go.
I see it.
And then we'll.
All right, so there's the link.
Awesome.
I like your mug, Alvin.
We get started or give people a couple minutes to join?
Yeah, let's right now we've got 4 people watching, it looks like. So 5, so people are starting to come in if we want to give everybody until maybe 1:30.
Yeah, that sounds good.
In 5 minutes. Do you want to play the stream of the pictures?
We're gonna do it at the end.
Okay.
But what I can do is I will actually start the intro comments, just have that up.
Yeah, that works. And if anybody who is joining now has any questions that they want to put in the chat, we can kind of start collecting those for the Q&A.
Yeah. Can you enable screen sharing?
Sure. You should be able to hit the screen share button at the bottom, or let me, all right. I've just made you the host, Kevin, so you should be able to do that. And then if you need to have somebody else share their screen, you can make them the host. And for the folks that are just coming on now, thanks so much for being patient with us while we get all this together. We're going to give everybody another minute or two, and then we'll go ahead and get started.
Let's get started. There's 17 people on the call, I think.
Yeah, it looks like it bumps around, so let's go ahead and get started.
Great.
All right.
So, hi everyone. Sorry for the technical difficulties, but thank you for joining us today. Just before we get started, a couple of brief announcements. So this is our 12th meeting. Here's our board of directors. They were newly voted in November of last year. So if we could have people just kind of go across and introduce themselves briefly, that'd be great. I can start. I'm the president currently. I'm also an MD PhD student at Penn. I've been growing carnivorous plants for over 10 years now. Josh, you want
I
to
guess
go?
it makes it my turn.
Yeah.
Kevin, first off, I told you to get rid of that awful picture with the sun in my eyes. I look like I was drinking something that was not on the table there.
You didn't send me a better one.
You could screen capture me, it would be better. That is the worst picture. I'm Joshua Weigert, the one with the terrible picture. I've been keeping all sorts of different kinds of carnivorous plants for years and years. The vice president for what, 3 years, 2 years, whatever. And I guess I'll move it on to the next person. Also, Jackie, you're being eaten by your background.
Oh, I know. I'm sure I am. I wasn't sure. I just did it for a laugh, but you know. I'm Jackie Bowman. I have been growing carnivorous plants probably for about 6-7 years. I'm the treasurer, and in my day job, I work at the Mutter Museum in Philadelphia.
Hi, I'm Anna Mim. This is my second year as the secretary. I've been growing carnivorous plants for like 3 or 4 years. In my day job, I work in public health.
Hi, everyone. I'm Alvin. I'm an undergrad at the University of Pittsburgh, and I've been growing carnivorous plants for about 10 years now. Yeah, that seems about right. And I've been with MACPS for 2 years now as a director.
Is Christine on?
Christine is in the YouTube one. I don't think she was
Gotcha.
able to get back in.
All right. So my name's Jamie Algier. I've been a director. This is my second year. I've been growing carnivorous plants for, I don't know, 4 or 5 years, something like that. My favorite genus is probably, I don't know. We'll go with Pinguicula today. It really just depends. And in the rest of my life, I am currently back in school to go finish up my master's degree in microbiology and just working towards that. So
Thanks. Rick, you just joined. You want to introduce yourself?
Yeah, hi, my name is Rick. I been growing carnivorous plants for probably near 10 years now. I've grown a little bit of everything, but I specialize mostly in the Nepenthes. Outside of that, I'm a nursery manager at a plant nursery, and I've been doing that for several years now. And that's pretty much it for me.
Awesome, thanks. So in terms of the schedule for today, we are running a little bit behind, but we will start with a carnivorous plant Q&A. I think we can make this a little bit shorter because we did start a little bit late. And then Dr. Tanya Renner, who's on the call, will be giving our keynote speech. Following that, we will have a presentation on Drosera of the New Jersey Pinelands given by Alvin Liu, and then we'll close out with a photo show. A brief reminder for those of you who don't know, we do offer memberships. Memberships come with the benefit of selling in our plant auction. We occasionally will host an auction of special specimen plants. And so if you're a member, you can participate in these. We have subsidies when applicable. For past field trips, we've given hotel subsidies. As a member, you also get a 10% discounted carnivorous plant nursery. And then we have members only meals or events. We have a couple of types of memberships you can see. Individual memberships are $20 a year. Family memberships are $30 a year. And if you're a student, you can get a $5 per year membership. And just visit our website, macpsociety.com, to join. For membership that was purchased in the end of 2020 or in 2021, you will be covered for 2 years until December 31st, 2022. And the reason we made this is because this past year and this upcoming year, we're not sure how many in-person meetings we'll be able to have. Some upcoming meetings to put on your calendar. The next one will be April 17th of this year. followed by July 17th, and then September 18th. We're hoping to have an in-person meeting later this year if it is safe to do so. So that is all. With that, I'd like to open it up for the carnivorous plant cultivation Q&A, which Anna will be moderating. So if you have any questions, just put them in the YouTube chat.
So we have a few questions that were submitted ahead of time, so we can start with those. Somebody said, not exactly carnivorous plant related, but how do you guys get super lush sphagnum moss to grow in terrariums? Does anybody have any answers for that?
Certainly
So-
over high humidity, I would say, and using rainwater or RO water might be a good idea. That would be my 2 suggestions.
Yeah, I would say humidity is definitely the biggest factor. It helps to have a little bit of airflow because mold seems to be an issue that inhibits its growth. So you want to make sure there's a little bit of airflow. I also find that it doesn't grow particularly well if it is incredibly hot. So if you've got something in the 80, 90 degree range all the time, it doesn't grow as well as it, for me at least. If it's slightly cooler, but really high humidity definitely helps.
I actually don't grow sphagnum moss. There's lots of other kinds of moss that'll grow a lot easier in most terrarium setups and most plant setups. I take from my aquarium Java moss, throw it on top, and it'll turn dark green and keep growing immersed without any problem and looks really, really nice.
I've been experimenting with a new product from Dynagrow called Orchid Pro. I've been using that in my greenhouse, so it does depend on what you're growing around the moss or with the moss, but I have noticed a substantial increase in growth rate and also vibrancy of color with that. So you might give that a look. You just dilute it to the minimum dilution.
All right, those are all really good answers and suggestions. Somebody else says, I live in an apartment in New York City where my Venus flytraps and Sarracenia have been indoors by a windowsill at room temperature. They don't appear to be going through dormancy. I have access to an outdoor balcony. My question is it too late in the season to take them outside? I've been scared of having my plants freeze in the winter, but I wonder if it would be better for them to have at least some dormant period this season instead of none at all.
Yeah, I would definitely say it's too late. So if you're going to try to force a plant to dormancy now, it's probably not going to do very well. There are even reports, questioning whether Venus flytraps even need a dormancy. If you grow them indoors under very high light, they might be able to get by without dormancy. That does not hold true for Sarracenia. The best thing you can do, so a windowsill may not be bright enough. I would give it very, very bright light. So if you have any kind of artificial light that you could put over it, that'd be great. But yeah, just maintain it, kind of assume that it's going through its growing season right now.
This is actually going back to the first question. Somebody in the YouTube chat has asked if subwassertang would work as like a moss. Josh, do you have any experience with that? Or anybody else?
Savassertang, you're going to have a really interesting problem with. It's not actually a moss. Savassertang is a life stage of a fern. And when you keep it underwater, it grows and it looks like a moss. When you take it out of the water and plant it in soil or in sphagnum or something like that, it's actually going to develop into ferns that look like little Boston ferns. They're kind of neat, but you're not going to get the effect you want.
Somebody is asking specifically about Sarracenia purpura. Do I cut off the half-dried pitchers when dividing and repotting in the spring, or do I leave them there?
I personally like to cut dried pitchers back. I find that I have less issues with problematic insects if I get rid of a lot of the litter, leaf litter, dried pitchers, things like that. But I think at the end of the day, the plant doesn't really care either way.
How long do pictures last on Nepenthes?
I think it varies, especially varies depending on how you grow them. But generally, I would say at least a couple of weeks. Sometimes pictures can hang there for 2 months, I would say.
I've had pictures on one of my plants, the same picture for over a year. So it just depends. I had happened to take a picture and somebody asked this in one of the plant forums online, who told me they can't last more than 6 weeks. So I went and I took a picture. It was a little over a year later. So I think it just really depends on the conditions. As long as the plant's healthy and the leaves are healthy, they can last quite a while.
My lowland Nepenthes have all stopped pitching, but the sundews in the same tank are thriving. What variables should I adjust? Maybe humidity is too low, maybe the night was too short at 4 hours.
I don't think the night length is going to matter so much. It matters for flowering, not for picturing, I believe. But if I'm wrong, someone please correct me. Usually, a Nepenthes that doesn't picture, you can look at either light as a factor or humidity as a factor. So the first thing I would try to do is up your humidity. And the second thing is also to add more supplemental light.
We have a question saying, I'm interested in field trips when we could do this sometime, maybe the Pine Barrens. And we do normally have one field trip in the summer. We didn't last year and we probably won't this year. As long as it's safe, we will. And I know we had talked about doing the Pine Barrens as well as a few other locations. Another question, what recommendations would you have for a carnivorous plant beginner to grow outdoors in this region with little effort? I grow all kinds of Sarracenia outside and Drosera filiformis is easy. Any others?
I suppose we have to know what region the person is in.
Yeah.
I think for this region, a lot of fly traps can also be easy, but with the stipulation that dormancy, they don't take dormancy as well as the Sarracenia, I think. So if you really heavily mulch fly traps, I think they can work pretty well, and they're really easy in the spring, summer, fall.
I keep some Rotunda flora and intermedia outside as well. Or rotundifolia. Sorry.
Is it normal to have some nectar bead on the outside of a Nepenthes pitcher, or is that indicative of a problem?
No, I wouldn't say that nectar production is any issue. Actually, I think that's actually a good sign in most cases. Usually nectar production all over the pitcher indicates that it's doing what it's supposed to do and that it's happy. The main thing that you would want to look out for is to make sure that it doesn't develop into any kind of sooty mold. Sometimes if you don't have enough airflow or something like that, the nectar can cause some issues with that, but it's not usually a big problem.
Oh, and back to the question about beginner plants to grow in this region. The person says they were being asked by somebody north of Baltimore, so in like Maryland. I don't know if that changes anybody's answer at all.
Same answer, I think.
Yeah. All right, I think those are all the questions we have. I'm pretty sure I got them all. And it's like almost 1:45, so that's probably a good time to move on as well.
That sounds great. Thanks, Anna. So I'm delighted to introduce Dr. Tanya Renner, an assistant professor of entomology at Penn State. Dr. Renner graduated from the San Diego State University and earned her PhD in plant biology at UC Berkeley. Her research uses plants and insects as model organisms to study the evolution of functional diversification and she is particularly interested in understanding how organisms are able to build upon existing genes and structures to gain novel phenotypes. As a member of Penn State's Center for Parasitic and Carnivorous Plants, Dr. Renner has extensively published on the genetics, biochemistry, and evolution of carnivorous plants. And we here at MACPS are delighted for her to give the keynote speech for today's meeting. Thank you, Dr. Renner.
Yeah, thanks for the invitation. It's really fun to be able to finally do this. So I'm going to go ahead and share my screen. Is that okay?
Yeah.
Okay. Oh, it looks like it's host disabled. I can't do it.
Oh, 1 second.
Do you know how to do it, Kevin?
You might have to make me a co-host.
Yeah, I'm gonna do
Yeah,
that. Right
if you click her name on the participant list, you can choose to make her a host.
There you go.
All right, let's see. All right. Can you see this?
Yep.
Okay, great. So yeah, thanks for inviting me today. It's so exciting to hear about all of your backgrounds, and I'll give you a little bit background into how I got into studying carnivorous plants too. But today I'm going to go into some of the work that we've done on studying the evolution of structural and chemical defense in carnivorous plants. And I like to think of carnivorous plants as being sort of like natural mutants to study plant physiology. It makes us, being able to study them allows us to be able to understand what's going on in other plants as well. And Kevin was mentioning, yeah, I'm an assistant professor of entomology. I study a lot of plant-insect interactions as well as some work with just insects. And I'm also a faculty member in the Huck Institutes of Life Sciences, and that brings together many plant biologists in various departments at Penn State. And we recently started the Center for Parasitic and Carnivorous Plants, which brings together many different faculty that study these amazing organisms that actually tend to have a number of features in common with one another. So that's been really fun. And hopefully when we get to be back on campus and doing more research, we'll be able to continue collaborations related to that. And you can see we definitely have to bring Rafflesia together with butterworts, and butterworts are one of my favorite too. So just to give you some background on myself, Kevin was mentioning my BS is from San Diego State University, and it's there I really started loving the study of molecular evolution. So like how do genes and gene families change over time? You know, what makes a protein do what it needs to do? And I started really studying this in liverworts and other relatives and thinking about how do plants deal with stresses, especially when they're coming up onto land, the history of land plants, and dealing with all sorts of stresses like heat and light. And then I did a little stint working in Ollie Ryder's lab at CRESS, which is associated with the San Diego Zoological Society. And I did some California condor genetic work. That was fun. And then I got back to plants working with Chelsea Specht at UC Berkeley, and now she's at Cornell University, so she's pretty close. I can get in the car and see her. And this is me working with an undergraduate, Sarah Starkey, looking at some plants at the UC Botanical Garden, which has a really nice collection of carnivorous plants. And there's also a really cool carnivorous plant society associated with the Bay Area too. So I've done some talks for them in the past and some work. And then this is actually California Carnivores, if any of you have ever had a chance to be there. Peter D'Amato, I think is a co-owner for that still. And this is me and Chelsea in these kiddie pools looking at Sarracenia up close. That's fun. And then after I was at UC Berkeley, I started working on some insect chemical defense with these really cool beetles that have unique associations with ants, and then some other cool beetles like bombardier beetles. And that work was with Wendy Moore at University of Arizona. So if it's hard for you to see a theme between all the things that I've done, I'm really interested in how organisms protect themselves, whether it's like against abiotic things like stress, think non-living stress, as well as living stresses. And I still do quite a bit of work on carnivorous plants. It's one of our main foci in the lab. So what do we do? How do we study the evolution of chemical and structural defense in many different organisms? Well, one, we have to be able to understand where some of these genes or proteins are present in an organism's body. And so we do a lot of studies on morphology, looking at cool structures underneath the scope. This is a gland here in an insect. We also do some comparative genomics where we sequence genomes and then also look at the genes that are being turned on in certain parts of the body to figure out, well, how is this plant or this insect actually able to do what it needs to do? And then we tie that together with chemistry, one looking at chemical compounds themselves, protective chemical compounds, as well as some proteins like in the carnivorous plants that are used to digest insect prey. And then we tie this together with thinking about the evolutionary history of, say, those proteins that are involved in defense. And this is just a gene tree showing the relationships amongst a variety of different chitinases in carnivorous Chirophilili. So that includes like sundews and fly traps and nepenthes. And we'll get into more of that in detail. So we kind of bring a variety of different things to the table to be able to study them. But we also like to get out in the field, because how do you be able to study things that are happening at the genetic level without understanding where they come from? And so we've done a variety of different field trips. This is the Green Swamp near Supply, North Carolina. And this was a fun trip that I took with an undergraduate of mine. And it was really cool. It's the first time I've ever seen a Venus flytrap in the wild. And I think what was so cool to me is just these longleaf pines that were in the area and just the amount of longleaf pine litter that's on the ground. And I was also with a group of entomologists, and they're, you know, we expect to see like so many different, so many fly traps in this area. And they were like, where are they? We can't see any of them. And I was like, they're everywhere, because they're flowering, and you could see their flowers like little white flags above all of this longleaf pine litter. And so you can see some of this. This is a longleaf pines needles, and then they were flowering like this, really high above their traps. And in some cases, you have to like push the needles away to even be able to see the traps. So that was really cool. And there was also some Sarracenia flava there, and some Drosera capularis, the pink sundew. So that was a super fun site. But we also have sites that are about 20 minutes away from the Penn State campus. Oh wait, I have to show you more pictures of fly traps before I get there. So this fly trap is really cool. So you can see the flowers, they're super white. And then in some cases, we did find some traps that had holes on the side. So some captured insects were making their way out of that situation, which I thought was pretty funny. But in our own area, we have some areas that are inundated with water, and they kind of sit there, and it makes for a boggy area. And one of our closest sites is Black Meshannon State Park, which again is about 20 minutes away from the Penn State campus. And you can see here from this picture, there's kind of like 3 fingers to this boggy area. And the water looks like tea because as the leaves are dropping into the water, they sit there and they get dark and look like black tea. And that's why it's called Black Meshannon. That's where the name comes from. But similarly, in that area, there are a lot of really big trees, even pretty close to the bog. So it's pretty much dominated by hemlock and white pine, oaks and black cherry and red maple, and just makes for a really interesting area because you'll be walking out towards the water and you see all sorts of blueberries too and rhododendron relatives, and you're like, where are these plants? But in fact, it's kind of nice that they put a boardwalk here because it protects the plants that are in the area. And you can see blueberry that's sitting right here, and these are members on my lab, past and present members on my lab. And we're looking down over the boardwalk, and you can easily find Drosera rotundifolia. There's Sarracenia purpurea, which you can see really easily when they're flowering during the spring and summer. Sometimes you can't find them without seeing those flowers that are held so high above the traps. So yeah, so we get out and we see these plants too. We're not just lab rats. So I want to mention that. It's fun. But just before I get to talking about, you know, what exactly are we studying, I want to give kind of a historical perspective and talk about some things or some features that we think binds carnivorous plants together. Like what makes a carnivorous plant carnivorous? So you have to go back into the literature and see first, you know, who were studying carnivorous plants to begin with. And this goes back pretty far. Charles Darwin had an entire book on carnivorous plants, was called Insectivorous Plants. And Darwin loved these plants. He would do all sorts of experiments with them. And this is just going to recount about how cool this system was and how it found that they were secreting, at the time they didn't really know what it was, but secreting something that would break down whatever they put on the plant. And Darwin put all sorts of interesting things on these plants, like cheese and meat. And you got to do a lot of experiments as a scientist. And we can all be scientists, right? And be able to do these types of experiments too. But what's also kind of funny is this is kind of a weird thing to think about plants eating other organisms, right? It's kind of like backwards to anything that we would be able to understand against nature in a way, right? Like we should be eating the plants shouldn't be eating us. And there's a quote out there from Carl Linnaeus, who's like the father of botany. And he was kind of against this idea of like to think that plants ate insects would go against the order of nature as willed by God. Like this is a really weird thing that these plants would be eating organisms, and that can't possibly be true. So since Darwin and Carl, we've learned a lot more about carnivorous plants, and folks have actually linked back the enzymes they found associated with the digestive fluid to the plant, that the plant is actually making it. It's not like something that is living with the plant. In some cases, that is the case. But for many carnivorous plants, we know that the plant is actually doing the digesting. They're doing a lot of the job. And sometimes they get help from some friends. So let's look at plant carnivory and what binds them together. So we know so far that the ability to digest prey or detritus or get nutrients from an external source that is not from the root system, not coming from the ground and uptaken by the roots, has evolved in at least 5 orders. And we think at least 10 times within those 5 orders. And there are about 800 or so species of carnivorous plants, which is pretty wild if you think about just the amount of diversity, right? And what's also really cool is that they have convergent morphologies. And what I mean by that is that you get similarities in the types of ways the leaves have been modified to trap prey. So for example, Pinguicula and sundews, we know, have like adhesive leaves that kind of act like flypaper traps. And then we have great examples like cephalotus and some really cool monocots that are carnivorous, like Burkinia and Catopsis, that have these tank-like structures to trap prey. And they're also often living in nutrient-poor sites, and that the essential nutrients that are taken up by their modified leaves are primarily nitrogen and phosphorus. So there's all sorts of cool studies to look at whether or not a plant is uptaking nitrogen and phosphorus from prey using like heavily labeled nitrogen and phosphorus, and looking at whether or not the plant has done something with those heavy compounds. And so there's some really cool literature out there if you're interested in reading about that. But I have to argue that there's probably 4 different attributes of a carnivorous plant. And of course, this gets pushed around, and even us folks that study carnivorous plants for our career still go back and forth on some of these. But I just want to go through 4 of them that seem to be linked, and many of us can agree that these are the things that link them together. So let's look at traction to begin with. So we know that many of these plants likely produce scents or compounds. I mean, we know plants in general make compounds, right? When you smell a rose, you smell compounds. Sometimes if you've ever interacted with one of those titanium arums, like the big arum plants that smell really like sulfur compounds that smell like rotting flesh, like they make all sorts of compounds. Plants are like the chemists of the world. We know that some carnivorous plants make compounds too, but this actually hasn't been studied in much detail. So there was a really cool study by Jurgens et al. About 10 years ago that surveyed a few different carnivorous plants to figure out, are they making compounds that might attract insects to their carnivorous leaves. So for example, Dianea makes a compound that smells like fresh cut grass. Some sundews make some rancid-like smells like nonanic acid. And then you can find other compounds associated with Sarracenia that kind of have banana-like smells. If you've ever had a Hefeweizen beer, you might be right, you might recognize the smell too. So isoamyl acetate, for example. So there seems to be some evidence that the traps themselves might make compounds that bring insects to them. And there also might be a division between the compounds that the the flowers that they make and the leaves that they make, which is pretty cool. Because one of the ideas is that these plants don't want to trap their pollinators because that would prevent them being able to pass on pollen and to reproduce. So there's one really cool study out there that I'm just waiting to get published because it's on bioRxiv, and it's by Winnie Ho and her collaborators, and she actually looked at floral and pitcher scent diversification in Sarracenia. And what Winnie found is that depending on the species, she also looked at some close relatives to Sarracenia, like Helianthera and Darlingtonia, but that the leaves and the flowers had different compounds. They're emitting different compounds, which kind of links back to that hypothesis that the flowers and the leaves would be attracting different organisms. But also within Sarracenia, that compounds differed from species to species. So those different species emit different scents. So this may lead to some division between the types of insects that these plants are attracting. So that's a really cool study. I hope that comes out soon. There's been one other really good study that's been done in Dionea muscipula, where they actually did some experiments where they fed the plants because they were really curious if the emissions change after feeding the plant, like if the plant is well fed, if it doesn't need to be fed anymore. And they did find some evidence for that, but we really need to follow up on these types of studies. And so we've been playing around with some of this in our lab too. We recently had a Helianthera that flowered in the lab. And this was the first time this plant has ever flowered in my lab, and I was really pretty excited about it. So you can see our lab in the background. And so we have a variety of chemical ecologists at Penn State that study floral and leaf volatiles. And so I called my colleague Jim Tomlinson up and his graduate student Tristan Confer, and I said, hey, let's try and collect some compounds from the flowers and the leaves. And so this is Tristan's setup where he's collecting volatiles just from the pitchers, and he's bagged the flowers because he had already collected volatiles from just the flowers. And we found some interesting results, and we're still working on this, so we're not ready to tell you about it, but it's something that we're excited about. And I just want to show you just like, how do you actually collect compounds from carnivorous plants? So that's pretty fun. Okay. So one of the other things in terms of attracting prey is that many of these carnivorous plants also have a reward system. In the case of Nepenthes, like that question that we got earlier about there being like drops of nectar associated with the pitchers, often that's used to bring in prey. And if you have lots of nectar associated with your pitcher plant, that's a really, really happy plant. It's getting enough humidity. It's doing exactly what it needs to do to try to attract prey. So in this case, this video and this image here comes from my collaborator Ulrike Bauer's work. And she found that even Nepenthes, even though they don't like take the top of the plant, which is the operculum, the little hood, and close it like you might expect just looking at it if you didn't know, that actually as the insects are eating some of this nectar, even from the underside, of this pitcher lid, that if rainwater hits the top of the lid, it'll actually fling the ants inside. So there's some really cool adaptations out there. So this kind of combines a nectar reward and then some really cool biomechanics to trap prey. So that's Ulrike's work. She's at the University of Bristol. And then some other studies that have been found that are also related to like rewards. So this is a Nepenthes pitcher plant in the Chapuis, and this is a shrew that's visiting this pitcher plant, and it's also eating a reward that the plant has made. And cool, in the process of sitting on this pitcher plant, the shrew's here long enough to defecate into the plant. And so we think that this could actually be a really good source of nitrogen for the plant itself. And I always think this is like a really rude shrew in that it doesn't put the lid down on the toilet when after it uses it. And so it's a really cool interaction. And so this is just to get you thinking about how interactions between carnivorous plants and other organisms are not always just with insects. They can also be with other organisms. And then these plants often have, a lot of coloration associated with them. We'll see a lot of red coloration, but it turns out that most insects cannot see the color red. There's some groups that can, but not all of them can. And there was a cool study that came out by Krupp in 2013 that actually found that some carnivorous plants actually emit fluorescence. And it's associated with wax crystals around the opening of, say, these pitcher plants. So these are two distinct related pitcher plants. This is Nepenthes, this is Sarracenia, totally different orders, but convergently, they have similarities in terms of having wax near the opening of their pitcher and also some fluorescence. And we do know that insects can see fluorescence. And so this might help to drive insects to the traps too. So this is probably more important than the colors themselves that we can see. Okay, and then they also have to have a way to be able to capture them. And much of our work has been looking at the glands of carnivorous plants and their morphology and what do they look like inside. So some of the types of glands that you may be familiar with are stalked and sessile glands on adhesive-like traps, active traps, which also have sessile glands as well, and pitcher-like traps that we know are involved in trapping prey, and it's pretty hard to get outside of them. And so I want to talk a little bit more about sessile and stalk glands and how we think that this has evolved, and then we'll take a look at our work on pitcher plants. So some of the work that I had done early on was looking at the evolution of these glands that are in the Chirophyllales. So this Chirophyllales is the order of carnivorous plants that contains pitcher plants like Nepenthes, also sundews like Drosera, and Dionea, the flytrap, but also some other strange carnivorous plants that we probably don't have in our own collections. They're pretty rare, like Triphyllum, which is a part-time carnivorous plant. And some of us might have Drosophilum too, which is pretty cool. Really neat plant. And so we think that maybe these ancestor had some sessile glands that didn't have any vasculature associated with them. And when I say vasculature, I'm talking about the internal conductive tissue that would move sugars and potentially digestive enzymes, as well as water through that actual gland. And it's kind of weird. Some carnivorous plants like sundews and Triphyllum and Drosophilum do have this vasculature. And it makes you wonder, okay, well, normally plant hairs, like if you look at a non-carnivorous plant, normally plant hairs do not have vasculature associated with them. So it might be that the glands that we see that move, like say on the sundews, those stalked glands, could actually have evolved from leaflets, and they might just be small modified leaves that have now become this stalked gland. And so one of the works that we did was just characterizing all the glands that are in carnivorous plants and relatives in this order Chirophyllales, and we think maybe there could have been two ways by which we got stalked glands, maybe through pinnation, where you're taking those leaflets and modifying them a little bit, or maybe some emargination on the edge of the leaf that kind of make these little nubs, and then those could have further evolved to become stalked glands. And we also have some losses of stalked glands, like in Aldrovanda, the waterwheel plant, and Dionea, which just has sessile glands. And then some other really cool stuff happened in Triphyllum and Drosophilum in terms of having certain types of vasculature. So it's really pretty complicated. Being a carnivorous plant is pretty complicated, even down to the gland level. And so we've been continuing to study this, and we're just really interested in glands, especially because they are what exudes the digestive enzymes. And so we've also continued looking at some cool structural adaptations. This is just a picture of me at the Royal Botanic Gardens Kew after the ICPS conference, which I guess was in 2017. And so this is Nepenthes truncata, which is really fun. So we have done some Nepenthes work too. And some of this work stems from what we know about the waxes that are associated with these pitchers. And I just want to note that like all plants have waxes. They've got a cuticle, and it has lots of wax embedded, and these two other compounds called cutin and cutan. And then basically the whole plant is protected by this wax. And that's really important because it prevents loss of water. But then there's all sorts of cool adaptations that plants have done to basically prevent insect attachment. So I like to think of carnivorous plants as potentially taking this trait that all plants have and changing it up a little bit to help them trap prey. So I want to show you some cool pictures of this. So here's just a picture of Nepenthes that on the inside of the picture wall you can see is white, and all of that whiteness there is associated with waxes. So the white is the wax. So if you were going to look at this up close, underneath a really powerful microscope, you'll see that these waxes look like little scales attached. And what's really interesting about this is that if you touch it, they easily break off. And so folks such as Bukins et al. Published some really cool up-close images of insect feet that have touched these wax surfaces in Nepenthes, and that they can find is that there's waxes that attach all over these insect feet. And what ends up happening is that these insects almost ice skate on the picture wall. So they're not able to attach because the waxes have detached from the plant and are now totally stuck on the foot. So we wanted to look at this in more detail across different species of Nepenthes, because what's interesting is that some Nepenthes have this waxiness and some do not. And you kind of scratch your head and you're like, what is going on here? And what did maybe the ancestral Nepenthes have if there's so much diversity? Now we do know that different species of Nepenthes trap different types of organisms, and also non-organisms or like dead organisms like detritus, like falling leaves. For example, Nepenthes ampullaria traps falling leaves, and it gets, you could consider it maybe sort of a pseudo-vegetarian, if you will. So we want to study this in more detail. And some other things that were really interesting to my colleague that I worked with on this, so Rika Bauer again, she's a biomechanics person, is also just the structure of the peristome itself and how that's also really slippery, and the angle of it and how that might be involved in helping to trap plants. So we're looking at, before you take a look at these down here, which I know are the big images, we're looking at a really up close view of the peristome that's associated with the opening of the Nepenthes pitcher plant. And you can see that up close, when you look at this under the microscope up close, you can see that there's a lot of ridges. And if you look in between each one of these ridges, like in the valley, there's also overlying cells. And this also prevents attachment as well. It gets really slippery. And it acts kind of has a capillary-like action when the rim gets wet. So as soon as the rim gets wet, these structures can wick up water and then make it very slippery. So these plants have a very slippery based on the way that their cells are organized, but also those waxes that I talked to you about. It's like the perfect combination to trap insects. So we looked at the peristomes, but also the wax layer variation on the inner wall of the pitcher. And here's some great examples of that. So that first one I was showing earlier was full, like in this image in A. This is like a reduced, so like there's just a little bit of wax. And then for some species, it's totally absent. It's like we can't even really see tons of wax associated with that pitcher wall. And then we did some measurements of the peristome width and as well as the slope. And we took into account what we know about the relationships of the Nepenthes pitcher plants, and we mapped them on the species tree as we know it. So I'm going to show an image of the species tree, which is huge, and I do not expect you to be able to read anything really on it. But basically, we mapped the width of the peristome and the slope of the peristome and whether or not the wax is present. We were really curious, and you can see there's a lot of variation here across each of the species. Each one of these circles represents the species here. On the left is the peristome width, and on the right is looking at the slope of the peristome. And so we wanted to know, what did the ancestor have? Like, did they have wax? What did their peristome look like? And based on doing a study like this, we found that it's likely that having wax crystals and having very narrow symmetrical peristomes is probably what the ancestor had. And that wax crystals themselves associated with pitcher plants have been reduced or lost independently multiple times. So by distantly related Nepenthes seem to continue to lose these wax crystals. And it's kind of like, why? Why is that the case? And I think we really need to study more the ecology of these organisms, what they're trapping, get out into the fields and be able to understand what's happening, because most of our measurements were either in botanical gardens or personal collections. And I think we just don't know a whole lot about what these plants are trapping. And so I think we need to know that and be able to connect that with the morphology and the characters that we know about. Somebody's got to do that for their PhD or for their career, probably too. Take a long time. And one of the other features that links many carnivorous plants together is the ability to digest prey and to absorb the nutrients associated with that prey. And so we've been doing a bunch of work studying the evolution of digestive enzymes in different groups of carnivorous plants. But what's kind of cool is that we noticed by doing some protein studies of the digestive fluids that there are things like chitinases and amylases, which we have amylases in our mouth. There's proteases like we have in our stomach, lipases, peroxidases, you name it. They've got lots of aces. Anytime you see an ace, that's an enzyme. And so they've got a lot of these enzymes that are pumped out of these glands and onto the leaf surface or into the digestive fluid that's associated with the leaf. And then they have also got to have a way to absorb those nutrients and benefit from them. And of course, those glands are also playing a role in that. So the glands are both exuding enzymes and they're also involved in uptaking the nutrients after those prey items have been digested. So those glands have a huge function, play a huge role in carnivorous plants. And so I just showed some images here, really cool ones. This is a slice through Utricularia, so the aquatic bladderwort. And in here you can see these quadriphid glands that kind of look like little X's that are just found throughout the inside of this trap. And this image here is from a butterwort, so Pinguicula, and it's just studded with stalked and sessile glands. And you can see there's a ton of gnats that are just attached to them. They're like slobbering all the time in our lab too. So then it's kind of begs this question of like, why aren't all plants carnivorous? This is such a cool adaptation. You're getting nitrogen from your leaf system. You're not totally, getting everything from the soil. You've got another method to get nutrients. And the way I like to think about this is that non-carnivorous plants primarily get their nutrients from their root system. But carnivorous plants are in these areas that are really low in nutrient availability, but the plants themselves over time have become limited in their ability to uptake nutrients. So they're primarily getting their nitrogen and other essential nutrients like phosphorus through their leaf system. But when you go out into a bog, like some of those pictures I was showing you earlier, there's not just carnivorous plants there, right? And those are low nutrient areas. There's blueberries there. We saw there were some really big trees that are like a little bit away from the bogs, but they are pretty close. Now, those plants are likely going to have other adaptations to deal with low nutrients. So this is just one adaptation out of many to be able to get essential nutrients from the leaf system, like from other sources. And so they're kind of like, I want to say like the pandas of the plant world, right? Like when we go in the bog and we see them, we're like, wow, this is like an amazing plant. Look at all these cool adaptations. And they are really cool. But it's easy to forget about the other plants that are around them that probably have just as cool adaptations. They might not just be as evident. Okay, cool. So let's look at some of the groups where we have actually studied digestive enzymes and what we've learned from studying them. So one of the groups that we've been studying this quite a bit in is in the order Lamiales, which includes a few different families that have carnivorous plants, but we've been really focusing on those in the family Lentibularaceae. So Lentibularaceae has cool plants like Pinguicula, which has those simple adhesive leaves, Gynlicia, the corkscrews plants that have those really cool underground shoots that look like they're spiraled, pretty much looks kind of like a leaf that's a Pinguicula leaf that's been rolled up and now looks kind of like a straw and has backward facing hairs. And Utricularia, which I showed an image of earlier, which are those aquatic carnivorous plants. And here's just some images. This isn't mine. This is one of Barry Rice's from Sarracenia.com that shows Utricularia underneath the water. And then this one here is from Utricularia in our neck of the woods. and just showing like each one of these bladders is just stuffed with invertebrates. This is a happy plant. And so we have Utricularia near us too, but we've been really studying one particular species of Utricularia called Utricularia gibba. And so this is Utricularia gibba, and gibba is really unique in that it has an extremely small genome. And you go like, how do we compare that? Like, how do we know it's got a small genome? Well, through a variety of different methods, we figured out it has a really tiny genome, and it's a really, really tiny genome for a plant genome. So in comparison, there's a model plant that a bunch of people focus on as part of their studies. It's really cool. It's called Arabidopsis. And Arabidopsis has a genome that's about the size of 135 megabases. Now, Utricularia gibba is even smaller at around 101 megabases. And bases, remember, DNA is made up of multiple bases, and it's like how we are even talking to each other right now is based on our nuclear DNA, and then we also have some mitochondrial DNA. And plants are cool because they also have chloroplast DNA. But gibba is we consider to be like an example of an extreme genome because it's got such a small genome that it's very compact. And what that means is that all the genes in the genome are really close together to one another. There's none of this stuff that we call junk DNA that's like in between all the important regions of the genome. And there are some important things that are associated with junk DNA, but Utricularia gibba has seemed to kick out a lot of that. And it's also kicked out a lot of genes that it doesn't need. So for example, root-like genes, it doesn't have roots. If I should go back to that picture on the previous slide, it's really just floating around in the water. It doesn't need to attach to anything. And it seems to have lost a number of these types of genes. But what's really cool about this bladderwort genome is it's made lots of copies of genes that it really needs. And I put this image here before we get going. I start getting excited about the genes, but we use this method that's is a new type of way of sequencing genomes called PacBio, and it allows you to get really long pieces of DNA off of the machine, and that makes it really easy to be able to assemble the genome, or easier. And so we were able to get, in some cases, from one tip of the chromosome to the other side of the tip, and that's really difficult, especially for plants, but having a really small genome makes that easier. So let's talk about the things that it does have. So what seems to be really apparent in the Utricularia genome is that this plant has copy-pasted genes that are important for metabolic activities, like carnivory, in its genome. So it's gone and taken some genes like cysteine proteases, and I'll talk to you a little bit about cysteine proteases, and have just copy-pasted them one after another in the genome to have lots of copies around so that when preadin comes, it has all sorts of these genes ready to go to be able to turn them on and use them in carnivory. So that's pretty awesome to be able to see that at the genetic level. And so some of the types of metabolic functions that we saw, like lots of genes in their genome, are involved in like peptide transport. That'd be really important if you're getting those essential nitrogen and phosphorus into the plant body. ATPase activities, so like breaking down all sorts of different material, chitinase activities for making nicks in the exoskeleton of invertebrates, hydrolases, and even things that are related to possibly the movement of these plants, because they've got some of the fastest plant movement out there in terms of the suction-like way that it traps our invertebrates underneath the water. So we found some really cool things related to that. But I want to talk a little bit about these cysteine proteases that I mentioned. So cysteine protease, you probably don't see this on the side of the bottom when you're in the grocery store, but cysteine protease is basically like meat tenderizer. So we use it to tenderize like a steak, for example, a really hard steak, and you can buy it in the store and you sprinkle it on your meat and then it tenderizes it over time. Well, these plants have a lot of these cysteine proteases, these tenderizing proteins, but other plants have them too. So it was first described, this type of protein was first described in papaya, and it's called papain. And there was this really cool experiment that was done to look at the importance of cysteine protease in terms of protection against insects. So this study, he looked at control papaya, and they put some insects on the leaf, and they found that the insects did not develop very well. They were very tiny. They did not do well on these plants. But if they added an inhibitor for this type of protein, the insect did fine. It was like there was no cysteine protease even present. And when they washed the leaves of the latex that contained this enzyme, they did great too. And then the insects did well on other plants that didn't have any cysteine protease. So this was one of the first studies that really showed that this enzyme is super important for protection against insects. And many plants have cysteine protease, some don't. And what it's doing is it makes nicks in the digestive system of the insect, and that allows the insects to also get some sort of infection. So they're actually eating the plant material and uptaking the cysteine protease. So in carnivorous plants, we think this is doing it from the outside in a way. And so by having it in the digestive fluid, now if you uptake an invertebrate, that, even if an aquatic one, there's many similarities to aquatic invertebrates as there would be in an insect you find on land, that would help break down the prey over time. So Utricularia has basically a bunch of meat tenderizer inside of it. Okay. And so this is pretty complicated here, but I want to walk you through it. So we've been studying the evolutionary history of these cysteine proteases in Utricularia. So this is just a tree that shows the relationships of all the cysteine proteases that we know about in Utricularia. There's a lot you can see. And we're looking at those relationships to even fly traps have cysteine protease too, which is pretty wild. Those are distant related carnivorous plants. But even things like tomato and grape and Arabidopsis, that model plant, has cysteine protease, but not as many as Utricularia. We also looked at where these genes are being turned on in the plant. So we had tissue from Utricularia from the traps, the shoots, and also the inflorescence, the flower, where the flowers come from. We found that in many cases, these copies of these cysteine proteases were turned on in the trap. So they were specifically in the trap, which is really awesome. And then we also looked at how maybe some of these that are in the trap are evolving. And so what we did is we did some protein crystallization studies, and we were looking at where have there been potentially changes in these proteins? And we're really interested if there were changes that were in the part of the protein that would actually help break down the prey. And it turns out that in many cases for Utricularia, if we're looking at kind of a model of the protein, there were changes that happened in the part of the protein that was really important for functionality. So it seems like in Utricularia, there's been a lot of changes, and maybe these changes are helping this plant be carnivorous. So that was pretty neat to look at. Okay, how am I doing on time? I'm doing okay. I got a little bit more about Caraphylales, if that's okay. All right. So we've been also looking at digestive enzymes in the order Caraphylales for a variety of different species. And our work has been a lot on these types of proteins that are used to make a nick in the exoskeleton of the insect, and they're called chitinases. And so we know, I showed you earlier, that there's so many different types of enzymes that we can find in the digestive fluid. But in the carnivorous plants that are found in that order Caraphylales, chitinases are really super abundant if you categorize all the proteins that are in that digestive fluid. And so I was really curious, what is the history of these chitinases, similar to the cysteine protease study I was talking about earlier? Where did they come from? And what's kind of cool about carnivorous plants is that there are so many different types of enzymes, but they all look really similar to these, what they're called pathogenesis-related proteins that are found in all plants. So all plants pretty much have a way to be able to protect themselves against insects, against different types of pathogenic microbes, and carnivorous plants seem to be using a lot of these types of defense-like genes for new functions involved in breaking down insect prey. So it's like you're taking defense genes and now you're using them for digestion. So I just want to point out that all plants have these things that carnivorous plants have, but carnivorous plants may have made extra copies of them, or they work slightly different, but they share a history with those that we find in all different sorts of plants. So many of these have been described in tobacco and tomato, for example. And so these are just some different examples. So involved in breaking down proteins and lipids and sugars, for example. So we've been really looking at chitinases, and they're found in all flowering plants. And the plants usually, when they use chitinases, they use them for defending themselves against fungi. And so fungi, in their cell walls, they have chitin. And what's really kind of crazy about the chitin too is that we also find it in the exoskeleton of an insect. And so one of our ideas is like, well, maybe these carnivorous plants have taken chitinases that are used against fungi, pathogenic fungi, and are now using them to protect themselves, or not protect themselves, but to eat insects. And so I'm just going to show, like, I'm not going to show a protein, but we're going to consider a protein kind of look like Pac-Man, because I always, when I look at protein, they look like Pac-Man to me. And so in this case, we just have a Pac-Man protein that's eating fungi before it becomes a problem for the plant. And chitin is chitin, no matter whether or not it's made by a fungus or an insect, it looks the same to a chitinase. And so chitinase would work very similarly. So yeah, it's found in the fungal cell wall, and it's also found in the exoskeleton of an insect. So what happened? What happened in carnivorous plants in the history of making a carnivorous plant? So we think what might have happened based on our studies is that there was a non-carnivorous plant ancestor that had a chitinase that was just eating fungus. And then at some time in the evolutionary history of carnivorous plants coming on the scene, there was a duplication of this gene that codes for this protein that breaks down fungi. And all of a sudden now you can have a protein that still has defensive functions. You got to still protect yourself, but also have some that can be used specifically for carnivory. And what's really cool about the Carophylales carnivorous plants is that if you put an insect into the trap, it will turn on just the carnivorous plant chitinases. So there's also a timing thing that's very specific for carnivorous plants too. So we've been looking at this in more detail in other gene families, but chitinases are pretty darn rad. Okay, cool. So, so what would happen here, like in terms of the evolution of these genes? So we think that the defense chitinases are under pressure to like get away and to evade their fungal inhibitors. So fungi will actually make inhibitors to plug up these enzymes. And so plant enzymes like have to always change a little bit. It's like an arms race to get around this and to be able to work. But those that are used in digestion seem to be evolving differently than to the defense ones that are used in defense, because there are no known inhibitors that insects make to plug these up. So these digestion kinases can just keep doing what they're doing. They work really, really well, but those used in defense are like constantly in this arms race to change. And we can see that at the molecular level. Okay, so I just want to kind of end talking a little bit more about what we know about other carnivores. So my collaborator, Kenji Fukushima, who was the lead author on the cephalotus genome that came out in 2017. This is a great paper because it's kind of the Easter egg paper in that it didn't just present the cephalotus genome, but it also presented proteome data. So the data that tells us what kinds of proteins are in the fluid for distantly related carnivores. So they looked at what was in the fluid for cephalotus, but they also looked at this distant related pitcher plant Nepenthes, sundews, which we know are pretty closely related to Nepenthes, and then also Sarracenia purpurea, who's in a totally different order in the Uricales. And what was really cool is that distantly related carnivorous plants, they will convergently secrete the same types of digestive enzymes into their fluid. And they have independently have picked, picking and choosing, I know it's kind of anthropomorphizing it, but have picked defense genes and are using those for carnivory. And that has happened multiple times over the evolutionary history of carnivorous plants. That's kind of wild, right? Like, so these enzymes are working very well for digestive functions across distantly related carnivores. So we've been looking at this in more detail, also in collaboration with Kenji for other groups, like studying sundews some more. We're just getting into looking at some butterworts too, and so I'm really excited for that research. And we do know that we have been looking at sundews and they do move in response to putting prey on the plant. And some of you have probably experienced this with your own collections. But we're also really curious, like what kind of turns on the digestive enzymes. And so we've been looking at how certain phytohormones like jasmonic acid can actually help to turn on some of these carnivory genes. And so we have a grant to look at the role of phytohormones and how they are regulating, turning on these carnivory genes. And this might be an old holdover from how jasmonic acid can be used to turn on defense genes. So we want to know, do those phytohormones still work to turn on carnivory genes, which will be fun. So the other things we kind of think about are also applications. So can some of these carnivory genes still perform defensive functions? And like if we took them and use them like for biological control, for example, could they be really good at controlling, say, plant pathogens and insect vectors? And there was I want to just end on this super paper that came out in 2019 that showed that if you actually take a chitinase from Drosera rotundifolia and you put it into tobacco, so you make a transgenic tobacco, it makes the tobacco more, it can protect itself more from fungi than it could with its own chitinases. So it seems like some chitinases, even from sundews, can have protective features that may be important for, you know, agriculturally important plants. And so they did some really cool studies where they like ground up the tobacco after they added the sundew chitinase and then looked at whether or not it was able to knock down the fungus. And it definitely was able to. So that's pretty neat. So I think there will be some definite applications of studying these enzymes in the future. So if you're interested in learning more about our research, you can check out our website, our Penn State website, rennerlab.org. We're also on Twitter at rennerlab. You can read more about posts and whatnot of talks that are coming up. For example, definitely do some more reading. Barry Rice's Growing Carnivorous Plants book is great. If you don't have that already, there's been a variety of public media that have come out that we've contributed to. And then also the New Carnivorous Plants book that about 50 or so of us contributed to that came out a couple years ago. There's also some really great podcasts out there. We recently at Penn State were interviewed for the rule breakers of the plant world, myself and some other faculty members, as it relates to the Center for Parasitic and Carnivorous Plants. And definitely get out there. You gotta be able to see your plants out in the wild. I know our Saracenia is probably sitting under snow right now and it's doing fine. It loves it. So I encourage you to get out there and see plants in the wild if you can. And with that, I'll just have some acknowledgements for folks that have worked on genome projects. It definitely takes a huge team to be able to do that. It's really also important for us to work with undergraduate students in the lab and be able to teach them how to work with genetics and carnivorous plants. Those folks with STARS or undergrads that have worked in the lab. And also the botanical gardens that we've worked with and folks we've gotten tissue with for some unusual carnivores like Triphyophyllum for some projects, and our funding from the National Science Foundation. And I'm happy to take any questions. I hope I didn't go too over time. Thanks.
Thank you so much. That was so interesting. I learned a lot, and it sounds like you're doing some really cool research. So we will be sure to post the links to your research on our Facebook page. That way members can easily get to that, because that was one of my questions, was like, how do we keep up with what you're doing? And how do we, you know, when you do publish about the helium flora flowers and pictures, like, I want to know what you found out. So we'll be sure to post that so that all of our members can keep up with what you're doing.
Cool.
Kevin, do we want to do some questions or?
Yeah, maybe one question.
Okay.
Dr. Renner, also, I can post the YouTube link that we're streaming on in the chat, because there are actually a whole bunch of questions, if you don't mind.
There is. Okay, I wasn't able to keep what I would have loved to see them. I'm sorry, I couldn't answer them in real time. Usually when I'm doing lectures like in person, I just stop and we just have a conversation during the middle of it.
No, not at all, not at all.
Okay, let's see. Oh,
yeah, we do have quite a few questions.
Okay, awesome.
Adam was wondering if you have any cool literature that you can recommend for people.
Oh yeah, so that Carnivorous Plants book that came out recently, I know that one's kind of expensive and it has a good number of articles in it, But really, if you can get your hands on like Barry Rice's Growing Carnivorous Plant book and Peter D'Amato's, oh my gosh, I'm totally blanking. Does anyone know Peter's book off the top
Savage
of your head?
Garden.
Which one? What was that?
Savage Garden.
Yes, Savage Garden. That is a great one. And you know, so I think there's also good information there that kind of gets you thinking about the science behind the plants too, not just growing them. So there's interesting anecdotes. So I think those are probably the most accessible ones that you'll be able to find that are still in print and also aren't too expensive, because sometimes, you know, academic texts can be a little bit expensive. And I'm hoping with the Carnivorous Plant book, I think there are some softbound books that you can get now. At first was in hardbound, and I think they're less expensive than they were in the past. But also just really good websites. I mean, BarrySaracenia.com. I know I keep talking about Barry's work, but Barry's done so much. That's such a great resource online, as well as the International Carnivorous Plant Society websites that have articles related to many different species, as well as the Botanical Society of America also has a carnivory page with information. So those, you know, if you have the internet, which hopefully you are because you're here, those would be free ways to get more information.
Cool, thank you. We'll do one more question just so we don't get too behind schedule. Are there any symbiotic carnivorous plant and fungal relationships that we know of, like how plants will exchange nutrients via the roots and mycelium?
Yeah, so I think the most that we know about is who is there. So in Sarracenia and Nepenthes, so Dr. Kadeen Gilbert, who's a postdoctoral researcher in my lab, he did his PhD on the microbial inhabitants of Nepenthes pitcher plants, as well as some other trait evolution stuff. And his work, he really did find a whole bunch of different what you could call like endosymbionts, like inside of the pitcher fluid that are living there and likely helping with the digestion. So the helping part and how it happens, we don't know too much about yet. And there's new methods to be able to do that. And actually, Dr. Gilbert has been starting that as part of his postdoctoral research. And he has Nepenthes pitcher plants growing in growth chambers on campus and has been doing experiments with them. But yes, so you can find all different kinds of microbes, including fungi, inside the fluid. And the question is like, how much and what exactly are they doing? But they don't seem to be pathogenic. So I'm going to bank on it being somewhat beneficial.
Cool. Thank you so much. Yeah, we really loved having you. That was so interesting. We really appreciate you taking the time on a weekend to come talk to us and teach us about all the stuff you're up to. After this, Alvin is going to be talking about his book. Would you mind switching so that he can host and share his screen and tell us about what he's been doing?
I can do that. Okay.
Thank you.
Make a host. Awesome. I think it's happened.
Yeah, I think that worked.
All right, I think it's working.
Yes.
Okay, perfect. Hi, everyone. If you missed the introductions at the beginning of the meeting, I'm Alvin, and I'm an undergrad at the University of Pittsburgh. I'm planning on applying to medical school this year, but in my free time, I am obsessed with carnivorous plants, and specifically carnivorous plants in my home state, New Jersey. This past year, with my co-author Daniel DiPietro, we published a book on native Drosera of the New Jersey pinelands with the Botanical Research Institute of Texas Press. So I'd like to start off by showing off this quote that I shamelessly stole from the first chapter of my book. Their very existence seems to defy the natural order of life in which plants are invariably subjected to predation by animals. There is a common notion that the exotic and alien nature of the plants dictates that they must also grow in exotic and alien places. So that's a mouthful for basically saying, in the public perception, carnivorous plants are these strange and exotic things that don't exist like in close proximity to you. And I think this is embodied by this picture of Audrey II from Little Shop of Horrors, the popular Broadway musical. But this notion is not entirely true all the time. And for example, here we have a Drosera intermedia with the dragonfly that's captured, and this is growing in a recreational lake in Ocean County, New Jersey. There are houses like a couple hundred meters away from this site. So if there's one thing I want people to take away from this, it's that carnivorous plants are not quite as alien as you might think. So here's the New Jersey pinelands. It's about 1 million acres in southern New Jersey that spans 7 counties. And here I have to make the distinction between the term the New Jersey Pinelands and the Pinelands National Reserve, because the Pinelands National Reserve is a legislative term that's used for legal reasons to denote lands that are protected under law, and there's restricted development in that particular area. But the New Jersey Pinelands as a whole refers to the Pine Barrens ecoregion in southern New Jersey, which is mostly composed of sandy acidic soils dominated by pitch pines. And over here, there's a characteristic lake in the Pinelands. 2 very common species we see are Atlantic white cedars and American white water lilies. All of the lakes in the Pinelands are actually artificial. They were created by human drainage of wetlands. And as a consequence, we also have a lot of shallow runoff of these lakes. And very frequently, we have acidic peat bogs forming in these areas, which are dominated by Atlantic white cedars and sphagnum. In these peat bogs, we commonly see carnivorous plants. For example, on the left, we have Sarracenia purpurea in Burlington County. If I go on the map, can I use a pointer? Yeah. Burlington County is this kind of wedge-shaped one right here, and Ocean County is over here. Those are the 2 counties we're going to be talking about the most today. And on the right here, we have Utricularia inflata in Ocean County. This was actually in a recreational lake that was bounded by people's property. So when I was wading into the water to get this shot with my camera, I saw people on the opposite bank looking at me like I was some kind of lunatic. So the first species of Drosera I'd like to start off on is Drosera filiformis, or the threadleaf sundew. And this is one of the most common Drosera found in New Jersey. Here we have a magnificent view of these vast fields of Drosera filiformis growing in sandy flats. And on the right here, we have Drosera filiformis growing in a bog in Ocean County. So ecologically speaking, Drosera filiformis is found in a variety of habitats. Most frequently, people associate it with sphagnum hummocks and bogs. So we can see here, this is an Ocean County bog where the Drosera filiformis is associated with these mounds of sphagnum that rise out of the water. But surprisingly, and this is something that people usually don't think about because it seems so odd, Drosera filiformis can also be found growing in these really dry and sandy habitats where people think carnivorous plants can't survive. And it's really remarkable, because these man-made trails in pitch pine forest are miles away from the closest water source. So first of all, we have to question, how did the plants even get there in the first place? And second of all, how do these plants survive? Well, these trails tend to be seasonally wet. There's a lot of depressed swales in these trails that collect rainwater in the wet season, particularly in early spring and late fall. And apparently that's enough to sustain these sphagnum outcroppings that also contain a lot of Drosera, like these Drosera filiformis here. The plants also seem to be able to better survive because they have deep root systems compared to other Drosera, like Drosera intermedia or rotundiflora. And as an example of the incredible diversity of Drosera in the New Jersey pinelands, we have here 2 newly described forms of Drosera filiformis that were just published last year in CPN. On the left is Drosera filiformis forma rubiflora. The plants look identical to the normal Drosera filiformis in every way, except for these brilliant red-colored flowers. And so far, this form is only known from 1 population in Ocean County. And as a funny story about that, I actually got pulled over when I was going to check out those plants. It was by a lake that was scheduled for housing development, but apparently they found the endangered Pine Barrens tree frog there, and the government sort of confiscated the land so that it could be placed under NJ Department of Environmental Protection protection. But they didn't post any signs, so when I went along in my silver Scion stick shift cube, I got pulled over by a cop who was curious why I was driving into a bog. So that was fun. On the right here, we have Drosera filiformis forma viridis. It's the anthocyanin-free version of Drosera filiformis, and this is also only known from 1 site in Ocean County, and it's actually been extirpated in the wild. This is the photograph of the only known wild plant, and it's since disappeared. Fortunately, the material is in cultivation. The next species I'd like to talk about is Drosera intermedia, also known as the bird's nest sundew. And like Drosera filiformis, Drosera intermedia shows a great variance in habitat preferences. Over here on the left, we have Drosera intermedia floating as an aquatic plant in an Ocean County lake. And on the right here, we have it growing in the same sandy trails as Drosera filiformis, except a lot of these plants have been seemingly killed by drought. So a big question here is how these populations manage to persist in these dry and sandy trails when they apparently are dying. Well, if you see here, you can see that the seed pods have already ripened. This photo was taken in, I think, late August. So that's when there tends to be less rain. In late summer, there's more drought. So the plants grow kind of as facultative annuals in these locations we've observed, wherein the plants will germinate in spring, mature quickly, flower, set seed, and then they might get killed by the summer droughts. But the populations can persist through seed banks created by the previous population of plants. Another very common site you'll see in the pinelands is Drosera intermedia associated with cedar stumps and bogs. I think these are some of the most photogenic occurrences of Drosera in the pinelands. And most frequently you'll observe that the Drosera are grazed a little bit above the water level. That tends to happen because the water levels are higher in winter and the seeds float. They have hydrophobic coatings and they tend to stick to things that are jutting out of the water like cedar stumps. So if you see any Drosera sort of dotting these cedar stumps, you can note where the water level goes up into in winter. Another interesting thing to note about Drosera intermedia and New Jersey in particular is that a lot of them have this peculiar stem forming habit. Normally Drosera intermedia grows as a rosetted plant, but you can see here that it kind of grows this long scrambling stem. We've come to the conclusion that this is associated with high water levels, but it's not entirely caused by it. You never see stem forming plants in the sort of dry habitats that we discussed earlier, but in wet habitats, you'll frequently see these plants forming these long scrambling stems that are presumably made to keep the growth point above the water level. And apparently when the water levels recede, the plants actually revert back to their normal rosetted growth. So you can see this plant over here has this long trailing stem, but after it was stranded on this branch and water levels receded in late summer, the new growth is now in this rosetted form. There's also a spectrum because a lot of Drosera intermedia grow as fully aquatic plants, and you can see here that this plant is kind of just free floating in water and it's formed this long scrambling stem in an effort to keep the growth point above water. But again, it's not universal because if we go back to this slide, we can see that these plants are kind of just floating on the top of the water in their normal rosetted form. So obviously there is a correlation with higher water levels, but we also think there might be some sort of genetic component going on here because not all the plants do it for some reason. And here's another odd behavior we found in Drosera filiformis. It's called false vivipary. A lot of questions I get about this are what is true vivipary, and that's when seeds tend to germinate prematurely in the seed pod. So false vivipary is when some sort of hormonal imbalance causes the seed, the flower stalks to develop incorrectly, and we get this weird amalgamation of flower petals and partially formed mature plants on top of the flower inflorescences. The behavior seems to be completely random. We did collect a couple of samples of plants who had been displaying this behavior, so we could possibly see if it's consistent or not, but we're still waiting to see if that turns out to be true. And if you actually cut off these deformed plantlets and propagate them separately, they grow into normal plants. So it's quite interesting. And Drosera rotundifolia is the third species of sundew that's native to New Jersey. And this is Charles Darwin's fan favorite. And we can see in these two photographs that they display different growth habits depending on the light saturation levels. We touched on different water levels and how they impacted the growth of Drosera and with the previous two species. And with Drosera rotundifolia, I'd like to also show off that light levels are a significant factor in how these plants behave. Over here, we can see, again, like Drosera intermedia, Drosera rotundifolia kind of likes to hug cedar stumps, and we can see that the plants mark the highest extent of the water line over here. We don't see this habit with Drosera filiformis, probably for a couple of reasons. Physically, Drosera filiformis is just a really tall and top-heavy plant. So you can imagine that if any plant managed to colonize these sort of cedar stems, it'd fall over pretty quickly. Drosera filiformis is also less tolerant of water than Drosera rotundifolia and Drosera intermedia. So that's probably another reason why. And over here, we have a Drosera rotundifolia that's snuck into a crack in a cedar log. I think this photo was from Ocean County. So now we can get into some of the fun stuff. There are also two hybrids of Drosera that are native to the Pinelands. The first is Drosera eloysiana, which is the hybrid between Drosera rotundifolia and Drosera intermedia. This plant was formerly known as Drosera belizeana, and I won't bore you with the details of exactly why the name was changed. Suffice to say, it's a long and complicated story. But the primary reason is that the original specimens were apparently mislabeled. So it was first described in the 1880s by a French botanist named Edmond Gustave Camus, and he collected material in a bog near Paris, I believe. And this is his original publication. And if you're looking at this photograph or drawing and thinking to yourself, that looks a lot like Drosera rotundifolia, you'd be absolutely correct, because when people looked at digital photographs of the herbarium specimens that he originally collected, it turns out they were Drosera rotundifolia, not the hybrid. So that's, in a nutshell, the primary reason why the name was changed. On the left here, here's a photo courtesy of Jason Sepka of Drosera eloysiana growing next to one of its parents, Drosera intermedia. You'll note that the hybrid is a lot bigger and apparently more vigorous than the parent. That's an interesting phenomenon that people have dubbed hybrid vigor, and people aren't exactly sure why it happens. And it doesn't appear to happen with every single hybrid plant, but when it does happen, it's pretty obvious. You can see these massive clumps of just the hybrid dwarfing the parent species. It's really incredible to see. Unfortunately, identifying the hybrid is not always clear cut, like in the previous example. Over here, there's actually a Drosera eloysiana right here. You can identify it by the more rounded leaf laminas compared to the intermedia parents on either side. But because this plant is so small, it's really tough to identify. Here we've created a key to help people sort of identify these species, but as you can imagine, with hybrid variability, it can get pretty tricky. For example, these are both clones of Drosera eloysiana that are popular in cultivation, and they're both from New Jersey. On the left is Dr. Franken Snyder's Monster, created by Ivan Snyder, who does a lot of funky experiments with Drosera. This plant came from material sourced from Burlington County, I believe, and Drosera eloysiana normally is a sterile hybrid, but Mr. Snyder treated the plants with colchicine, which is an alkaloid chemical that can induce polyploidy in plants, and mutated it so that it could actually become fertile and reproduce by itself. So that's a little interesting. On the right is Drosera Nightmare. That's a clone from Ocean County. And we can see that the two plants are a bit different in their growth habits. Drosera Nightmare on the right tends to favor the intermedia parentage more. You can see it has these long petioles that are more rounded and skinnier, whereas the clone on the left tends to favor the rotundifolia parentage more. Drosera ex hybrida is the hybrid between Drosera filiformis and Drosera intermedia, and it's actually only ever been recorded from the New Jersey Pinelands, and only a handful of times. It's extremely rare in the wild, and if you get to see it, you are very lucky. Here we have a big massive Drosera hybrida growing in Burlington County. The reason this plant is so rare appears to be because of the barriers to hybridization between Drosera intermedia and Drosera filiformis. First of all, Drosera filiformis and intermedia flower at different times. Filiformis tends to flower in mid-May to early June, whereas intermedia tends to flower in late June to July. So in places where we see this hybrid occurring, the common theme seems to be some sort of artificial disturbance. For example, one of the first populations of Drosera hybrida that was discovered was at Lake Absegami in Burlington County, New Jersey. It's actually where Peter D'Amato grew up. And the site was found near a recreational lake where a lot of lake goers would set up chairs and like swim and do stuff like that. And a lot of the Drosera filiformis at that site ended up getting trampled, which delayed their flowering and allowed them to actually overlap with the flowering period of Drosera intermedia. Here's another population from Ocean County. This population is from a trail, an off-country trail site that's used by a lot of ATVs and military vehicles going on patrols and drills. So the plants of that population get constantly run over and squished. So that's another factor that can delay the flowering of Drosera filiformis and allow it to coincide with the flowering period of Drosera intermedia. And this hybrid is a bit easier to identify than Drosera aloysiana, just because the parents look very different and the hybrid itself has a different leaf shape compared to Drosera filiformis. You can see that the leaf of Drosera filiformis is very tapered and kind of ensiform, while the leaf of Drosera hybrida has a more rounded edge. Now, another topic that I'd like to touch on, because it's really not discussed very frequently at all, is winter dormancy. We find that people really like to talk about how carnivorous plants look in full growth, especially because people are usually talking about Sarracenia in the southeast US, and we all know that those don't look very exciting in winter. But what do Drosera do? Well, most temperate Drosera form a specialized structure called a hibernaculum, which is essentially just a bundle of modified scale-like leaves. And the hibernaculum look different in every species. So on the left, we have Drosera filiformis. The hibernaculum of Drosera filiformis tend to be these sort of budded, circular, rounded bud structures, while the hibernacula of Drosera rotundifolia on the right here are more loose, and we can kind of see the individual modified leaves kind of poking out over there. An interesting implication is what this does for Drosera intermedia that form stems. It was unclear before if the stems of Drosera intermedia actually persisted throughout the winter or if the plants had to renew their growth every year. And it turns out the answer is that with Drosera intermedia, everything except the hibernacula dies at the end of each growing season. So we can see here that the hibernacula at the tip here is alive, but the rest of the stem, the lignified structure here, is clearly dead, and those will decay over the course of the winter. And this tends to happen a lot with aquatic populations. You'll remember that the stem forming habit is associated a lot with high water levels. So this is interesting in that it provides a potential mechanism for the plants to actually relocate, because once the roots in the stem deteriorate, these hibernacula are free floating and they can kind of just drift around to other areas of the habitat and sort of establish themselves in new areas. And over here, we can clearly see that the leaves of Drosera intermedia during dormancy are a little bit more loosely arrayed than Drosera filiformis, but it's still tightly arrayed into this specialized bud. Of course, I would be remiss to not mention that exploring is, among other things, an enjoyable enterprise. On the left here, I found a massive mat of Sarracenia purpurea, and I'm enjoying my newfound power. And on the right here, my co-author, Daniel, took a little swim by accident. And of course, as a kind and generous helper, I took a photo of him first before helping him out. And I'd like to acknowledge a lot of people and organizations that made the publication of this work possible, principally the Philadelphia Botanical Club, the Pinelands Preservation Alliance, the MACPS, and the Carnivorous Plant Society based in the UK. Over here, there's a list of individual contributors, including two of our own MACPS members, Jason Sepka and Stephen Nachajewski. Anyone has any questions, I will be glad to take them now. Hopefully the MACPS will be able to have a field trip this year, and we do intend on having a field trip to the Pine Barrens at some point. So this can be seen as a nice little teaser or a preview of that potential field trip.
Is your book available for sale, and where can people buy it from?
It is currently available for sale from the website of the Botanical Research Institute of Texas. I can post the link into the YouTube chat.
Yeah, that would be great. I know we had it on our Facebook page when it first was published, but let's get it out there again for everyone.
It's on our website as well. In the interest of time, we're going to move it along. So the last thing is the photo contest. Alvin, do you mind passing me host?
Yeah, just a second.
Thank you.
Thank you, Alvin, for sharing all your pictures and stuff. That was really interesting. It's so cool that you were able to find so many plants and write a whole book about it.
Awesome, thank you. So we had 81 submissions to our New Year's photo contest, so we'll play this and then we'll announce the winners at the very end. All right. Thanks to everyone for submitting. Rick, would you like to do the honors?
Yeah, I would. And I just wanted to thank everybody again for all the submissions and everything. I think we had a phenomenal turnout, and it was really great seeing everybody's stuff for sure. We're hoping to maybe make it an annual thing to have this as an event. So hopefully we'll get to do this again. As for the winners this year, we have our two winners are going to be submission number 2, which is Mr. Brad Lawrence. And the other one is going to be number 43 from our very own Alvin, of course. So congratulations to you guys. Additionally, we did want to make a couple honorable mentions. Mr. Adam Williams, number 24, got a real kick out of that one. Looks like the couple of flies getting busy over the Venus flytraps. And Keegan Orr, phenomenal composition on the Saraceni there with the lighting and the black backdrop, which is absolutely wonderful. But thank you guys so much.
Thank you, Dr. Renner. Thank you, Alvin, for those beautiful talks. And thank you all for listening. See you next time.
Thanks.
Thank you guys so much. Thank you, Dr. Renner. Thank you, Alvin.
Thanks, everyone. That was awesome.
Such great talks. Really good.